Hello, and welcome to G Fun Facts Online, the podcast where we dive deep into the most mind-bending facts from the world of science and technology. Today, we're tackling a topic straight out of science fiction, but it's happening right now in labs around the world. Imagine creating miniature human brains in a dish and then using them to power computers. Sounds wild, right? Well, get ready to explore the groundbreaking field of organoid intelligence, or OI. So what exactly is organoid intelligence? At its core, OI is about harnessing the incredible biological processing power of the human brain. Instead of silicon chips, scientists are using 3D brain organoids, tiny, self-organizing clusters of human brain cells grown from stem cells in a lab. Think of them as biological hardware. The goal? To create computing systems that could one day be faster, more efficient, and potentially even more powerful than the supercomputers we have today, all while using a fraction of the energy. These brain organoids aren't just tiny blobs of cells. They possess incredible biological properties like plasticity, the ability to adapt and change in response to new information, something that's fundamental to learning. They can process information in a fully parallel way, much like our own brains, and even show signs of anticipating things. Researchers have already taught these in vitro neuronal cultures to play the classic video game Pong, recognize speech, and perform simple math. There is even a system called BrainOware, which combines these organoids with traditional hardware, and it has learned to recognize speech and predict complex equations much faster than silicon-based systems. How amazing is that? To make this happen, scientists create a sophisticated setup. A 3D brain organoid sits at the heart, doing the computational heavy lifting. Tiny microfluidic systems keep it alive and healthy. Inputs can be electrical, chemical, or even signals from other connected organoids, like tiny retinal organoids that can see. Microelectrode arrays then deliver these inputs and measure the outputs, allowing researchers to model learning processes. And, of course, AI and machine learning algorithms are used to decode the complex data these biological computers generate. Now let's talk about the promises. The potential applications of organoid intelligence are simply staggering. In medicine, it's a game changer. These brains in a dish can mimic aspects of human brain development, making them perfect for studying devastating neurodevelopmental and neurodegenerative diseases like Alzheimer's, Parkinson's, or even the effects of viruses like Zika on brain development. Scientists can investigate human-specific brain disorders in ways that animal models just can't replicate. OI can also turbocharge drug discovery. Imagine testing new drugs or gene therapies quickly and safely on these mini-brain models, speeding up the development of life-saving treatments. And what about personalized medicine? We could potentially use organoids grown from a patient's own cells to study their specific disease, identify the best treatments tailored to their unique genetic makeup, leading to more effective therapies with fewer side effects. In the future, we might even see mature organoids used for tissue grafts to repair brain injuries. Beyond medicine, OI is set to revolutionize computing itself. It could unlock entirely new types of AI algorithms, inspired directly by the brain's architecture, potentially overcoming the limitations of current AI. We might even see carbon-silicon hybrid systems that combine the efficient learning of OI with the fast computing of traditional AI. Plus, OI offers a unique window into understanding the fundamentals of human cognition, how we process information, form memories, and make decisions. But, as with any groundbreaking technology, there are significant hurdles. Current brain organoids are still relatively simple. They contain hundreds of thousands of neurons, which sounds like a lot, but it's a drop in the ocean compared to the 86 billion neurons in a full human brain. They often lack proper vascularization, a blood supply system which limits their lifespan and complexity. They also miss some crucial cell types and the organized, layered structure of our brains. This means their processing power is still limited. Reproducibility is another challenge. Making sure organoids grown in different labs are consistent is tough. And developing sophisticated ways to talk to these 3D structures and interpret their signals is a huge engineering task. Not to mention managing and making sense of the sheer volume of complex data they produce. Researchers are working hard on solutions, developing assembloids, which are different region-specific organoids fused together, improving vascularization, and using advanced biomaterials. Now, this is where things get really deep. The prospect of creating increasingly sophisticated brains in a dish brings up some profound ethical, legal, and social questions. The biggest one: could these organoids ever develop some form of consciousness, sentience, or the ability to feel pain? While current evidence suggests this is unlikely, given their simplicity and lack of sensory input from a body or environment, the field is moving fast. If they did reach some level of sentience, it would fundamentally change how we view them and the ethics of using them in experiments. Then there's informed consent. These organoids are grown from human stem cells, often derived from skin or blood. Donors need to be fully aware of how their cells might be used, especially in research aiming to create forms of intelligence. What about implanting human brain organoids into animals to study them in a more complex environment? This raises concerns about animal welfare and the moral status of such chimeras. Clearly, we need strong ethical guidelines and regulations to govern OI research. And we need a broad societal discussion about the impact of creating new forms of intelligence. Even the term organoid intelligence, while catchy, needs careful handling to avoid hype and public misunderstanding. Thankfully, international collaborations are emerging, like the Baltimore Declaration toward OI, aiming to guide responsible progress. So organoid intelligence is at a truly fascinating crossroads. It holds immense promise to revolutionize neuroscience, medicine, and computing. Recent breakthroughs in 2024 and early 2025 are continually refining these tiny brains, making them more like the real thing, and integrating cool tech like CRISPR gene editing and advanced AI for analysis. We're even seeing more complex models being built by combining different types of organoids. The journey ahead requires a careful balance, pushing the boundaries of science while thoughtfully navigating the complex ethical terrain. It will take collaboration between neuroscientists, bioengineers, computer scientists, ethicists, and all of us, the public. Addressing the current limitations in organoid complexity and ensuring robust ethical dialogue are key to responsibly unlocking OI's transformative potential. What do you think about brains in a dish? It's a lot to process, isn't it? The future is unfolding right before our eyes. That's all the time we have for today on G Fun Facts Online. Thanks for tuning in. Join us next time for more amazing insights. Don't forget to subscribe and share if you found this as fascinating as I did.